autoimmunity. How many folks are working on autoimmune? Okay, good. All right, so the first question we're going to ask is human data available for multiple sclerosis, and what antigens have been mapped for T-cell epitopes. And then we'll look for therapeutic epitopes. So we tried to design these with as much real-life application. What people are actually interested in is epitopes that work and do something, either in the context of therapy, treatments, or vaccines, that sort of thing. So human multiple sclerosis. So we'll click in to get to the detailed disease search pane. We'll type in, we'll select multiple sclerosis. We'll then designate that we're interested in human data, just above. And I'm going to run the initial search just looking at that. Okay, so there's quite a bit of data, and antigens are always of great interest. Autoantigens are always of great interest, and there's a lot of, as I mentioned earlier, there is considerable overlap. There are some antigens that are very specific to a particular autoimmune disease, but there are also ones that overlap. So this gives you a list of quite a few that have been defined in the context of multiple sclerosis. And again, this is in the clinical context, so this is either EAE or humans with, actually this is, because I selected human, this is multiple sclerosis. So it just goes to show you there's a tremendous number of antigens that have been associated with multiple sclerosis. Okay, so the second part of that query was T-cell responses, so I'm just going to come back down here just to show you how quickly you can refine your search and hit search again. So we had 300 some total for the broad query. And by the way, we use search and query interchangeably, so if I'm switching back and forth between those two words, try not to be too off-put. We try to be consistent, but.
So the actual question asked for T-cell.
Did I? Oh, I did. Thank you very much. See, I did that just to see you were paying attention. Just kidding. I'm in a lull after lunch. See, you get an extra bonus. You get the B-cell ones too. Okay. So again, you see the list of epitopes here and a summary of the antigens from which these came. And then the third part of that query was, are there any T-cell epitopes that are defined in humans with MS that have been shown to be therapeutic? So I'm going to come down to the assay and type in treatment. And decreased disease is our generic term, our high node term for these set of assays. So I've further refined this particular search. There'll be far fewer, I promise. So there are two. So there have been two epitopes defined in the context of human disease and shown to be therapeutic. And I'll explain in a minute why the antigen and organism were blank. So I'm going down, I'm sorry if I'm going too fast, I'm going down into the details of those assays from the assay tab. I just picked the first one. So this particular epitope that was administered therapeutically to people was actually an analog of a natural sequence of myelin basic protein. So because the epitope itself is a synthetic, unnatural construct, so to speak. It was blank in the fields here. In case you were wondering where in the world these were blank. And that's not uncommon that synthetic analogs or mimotopes are used for therapeutic purposes, at least.
How did you see that it was an analog from that?
Pardon me?
How did you see it was an analog from?
So let me go back. So what I did is noting as I'm going that these are blank, my experience is telling me that these are probably analogs. So what I did is I clicked on the assay tab and then went into the details. So again, the proof is always in the details of the assay that we captured, because that comes directly from the paper. So that goes back to the original question we had about the non-peptidic epitope that was showing up blank. So I scrolled down to look at the epitope information, and sure enough, the epitope up here, the sequence that is the epitope, is shown in here, documented to be an analog of this natural sequence here. And so this is the key right here. Epitope is an analog of this natural sequence. And so these people with, so this shows occurrence of autoimmune disease, multiple sclerosis, chronic. They were administered this synthetic or non-natural epitope. In this case, they didn't provide details of how they administered it. Sometimes they do. Was found to be positive in 3 out of 5 individuals therapeutically. So treatment of epitope prevented relapse in some MS patients, although the outcome was not uniform. So not surprising, being a human population. All right. Any questions on that one? Yeah.
Other questions?
We don't curate that, and I can't remember in 10 years the time that I've annotated that. No, have you guys ever annotated that something was patented? Any curation? Okay, yeah, all right. Yeah, presumably if they find something therapeutic, they're going to patent it. Yes. Okay. So, another example. Oh, I will show you another example of the immunobrowser. So, are there immunodominant regions defined for insulin in humans? Defined along insulin in humans with type 2 diabetes. All right, so we're going to need the expanded version of the result page. All right, so we're going to look at human data, and we're going to look up diabetes. And this is the high node for diabetes. There are other related, for example, below that you see pre-diabetes syndrome. That's also captured for a lot of patients. It's also the designation used for some of the mouse models of type 1 diabetes, but just for simplicity, I'm just going to pick the high node of diabetes. Okay, and then I'm going to hit search. All right, so because I'm interested in the immunodominant regions, I'm going to use the Immunome browser. So to do that, I'm going to click on the antigen tab, and then I'm going to do something I told you can do, but I'm going to demonstrate it. So if you sort on the epitope, you can see the relative abundance of which are the heavy hitter, either in terms of responses or in terms of just being studied heavily, epitopes from GAD2 in this case, and insulin, and other antigens that some of you who study diabetes would recognize. In this case, here's insulin. So I'm going to go ahead and select the Immunome browser icon and click it. All right, and this is what it looks like when that gets graphed to the reference proteome for insulin in humans. And so you can see there's a couple of regions here of prominence and in humans. A couple of other areas that are more heavily studied is in terms of the number of assays. So you can look at those too. What I suggest is always when you generate these Immunin browser graphs, make sure you scrutinize the data within the table, because very often, let me show you an example. There is a region that actually scores very high. This region here, 44 to 51. So this actually had a very high RF score here. But because the regions around it, the longer peptides around it, the scores were not as high. The average along the graph is not reflecting that short peptide. So since this is, since these are, in this case, pause it down here. D1. So what I'm going to do, because it's a short peptide, and often with short peptides you have greater specificity versus 20 mers or 15 mers, I'm going to go ahead and peek at that and look at it. So these were T cell responses, and of course the length of the peptide is very important for specificity in T cell responses. So if we look at that, so there's the responsiveness is good, though the N is low. So while the overall number of people tested is somewhat low, the responsiveness is high. So what I'm getting at is, while the upper graph doesn't reflect that that particular peptide is a this particular peptide here is not looking like a prominent region or immunodominant region because it has such a high RF score, it might be something you'd be interested in looking at. So the devil's in the details. I would suggest always looking at the table and looking at the scores and comparing what actually has taken place by looking into the assay context there. And that'll get you a better idea. Okay.
Sorry, one question. If I select for disease and I want to know if what I found, the T-cell epitope also had relevance for other diseases, is there a filter function for that?
If you were interested in a particular epitope, probably the quickest way to do that is to copy and paste the epitope and into the linear search field here. If you copy and paste that epitope in there, in autoimmune disease, in the context of autoimmune disease, there's a chance that it could be involved in other diseases, correct? Maybe not the exact epitope, but certainly the antigen. So yes, you could copy and paste that in right here and just run a search and see what you can actually then look at the assay on the assay tab and scroll down on the immunization. Let me show you. If you ran a search for that, for a particular peptide, come here, and this column right here will show if there's an occurrence of disease. You also can type in, probably a better way to do it, I'm thinking on the fly here, is to type that and then select, generally just generically autoimmune, and see if it's in other autoimmune diseases. And similarly, you could do that for another type of disease. If you have a peptide that you want to see if it's related to other diseases, you could select the higher node or a more specific one there. Yeah.
But if you just want to know, if you take two diseases and you're looking for, common epitope for both of them, can you do that? Without
Yes.
knowing the specific?
Yes, because you can select. I mean, are you talking infectious disease? Or are you talking?
No, like autoimmune disease.
Autoimmune disease, yeah.
Like if it's type 1 diabetes. And
Yeah, so a quick way to do it is you could do it from the antigen level, and I would suggest you can run a query on that antigen of interest, the antigen that epitope is from, and then multi-select the different diseases that you're interested in, and run a query in that way. Bjorn? Yeah.
So, but there's no, I think what you're asking for is what epitopes are specifically found in two diseases and somehow specified disease A and disease B. And that's not how any of these work, unfortunately. So you'd have to do, if you currently select for two specific diseases, you'd get an OR query back. It must be in one or the other. And finding out which ones are in both, you'd have to do some kind of things like this manual.